Hi, it's Meg Carroll and Megan Nally, here to inform you about the medical world of 3D printing. Today, we'll be focusing on the use of 3D printed skin tissue through the process of bioprinting. Scientists at the Wake Forest Institute for Regenerative Medicine are the first in the world to successfully implant laboratory-grown organs into humans. The goal of their skin regeneration project is to bring the technology to soldiers who need it within the next 5 years. The loss of the skin barrier causes major fluid loss and increased risk of infection. Traditionally, extensive burns would be covered in skin from other parts of the body, but in extreme cases, there may not be enough healthy skin to cover the area. A printer has been designed to print skin cells onto burn wounds. The ink used is made of different kinds of skin cells. A scanner is then used to determine the size and depth of the burn. Data of the skin cell type guides the printer and applies layers of the correct type of cells to cover the wound. The skin regeneration technique is called bioprinting, in which scientists build an organ layer by layer using scanners and printers traditionally reserved for auto design, model building, and product prototyping. Bioprinters are very similar to inkjet printers, but have an elevator that moves a platform up and down, allowing three-dimensional printing. As each layer is added, the three-dimensional shape begins to form and develop. Here we can see a bioprinter in its major parts: the elevator, the platform, reservoirs, microgel, and bioink. Before the bioprinting process begins, a model must be created, along with a list of materials that will be used. The cells being used are then mixed with a liquefied material that provides oxygen and nutrients to keep them alive. This mixture is then placed in a printer cartridge. The cells are later dispensed onto a scaffold layer by layer to produce a tissue-like three-dimensional structure. Following skin transplantation, recipient T cells activate to donor antigens through three distinct pathways. The direct pathway, in which T cells recognize molecules on donor antigen-presenting cells. The indirect pathway, in which recipient T cells recognize donor antigens presented by self-major histocompatibility complex molecules on host antigen-presenting cells. And the semi-direct pathway, in which recipient T cells can be activated by acquired donor major histocompatibility complex molecules displayed on the recipient antigen-presenting cells. Bioprinting and organ printing are beneficial for multiple reasons. One, being it is an automated system making the mass production of tissues and organs possible, which will help save lives more quickly. It also has the ability to print numerous amounts of different cells. In addition, the printing is capable of occurring within the body itself, thus making it less invasive. There is a more than 119,000 person waiting list for organ donation, and 22 people die each day waiting for a transplant. In 2015 in the United States, 486,000 burn injuries received medical treatment, according to the American Burn Association. Many patients with major burns involving at least 25% of their total body surface die. The transplantation of large patches of skin would save their lives. The end goal of this research is to be able to 3D print all organs for clinical use. The effect would be huge, helping in clinical diagnostics, drug discovery, drug toxicity, and of course, human disease. In the next few years, scientists have predicted that technology will allow for the printing of all functional organs in the human body. This technology could save the lives of thousands of people at risk of death without a specific organ. Maybe, one day, you could receive a 3D printed organ. Thanks for watching!